you can include sites encoding protease recognition sites. Like here we have thrombin, but it can be also TEF protease, which is a viral protease, or precision protease. So just remember that you have your gene and you should remove your stop codon, put it in frame with the, glutathione has no stop codon apparently, and then you just clone your gene. You can use one of the restriction sites, which are, I think, in 3 different open reading frames. Usually we remove start codon from our protein, and then the gene, we will get a fusion gene and the fusion protein. And after purification, we can cleave our protein of interest off using the protease. And we have different types of bacterial promoters. Like here we have this P-TAC promoter, PTAC, which is a hybrid promoter consisting of lactose and tryptophan operons. It's still inducible with the IPTG as a lactose mimic, but it just yields a higher expression than the regular lactose promoter. The lactose promoter that we are using generally in the Escherichia coli system is the PLAC UV5 promoter. It contains, I think, 2 mutations in comparison to the wild type promoter, and it also yields a higher expression, which means that the bacterial RNA polymerase is recognizing this promoter and is binding with higher affinity, and we just get more mRNA. And then what you may also encounter is we have another tryptophan lactose promoter, which is TRP. It is just a different kind of a fusion between those 2 promoters. And they also differ bacteria in which they are active. So some promoters will not be active in some bacterial species. In E. coli, usually we have no problems, but if you want to express proteins in different bacterial species, for example, to study the function of some genes, then you have to pay attention to that fact. We have also arabinose promoter, which is PBAD, which is inducible with arabinose, and it's also its basal expression is suppressed with the high glucose. And we have also a phage promoter, lambda phage promoter PL, which carries a mutation making the repressor temperature sensitive. So by increasing the temperature of the culture, we can induce the expression. Those different types of promoter, they differ in the efficiency of expression, but they also differ in the degree to which they are leaky. Leaky means that you have some basal expression even without the inducing agent like IPTG. And unfortunately, our TAC promoter is a little bit leaky. If we want a system which is a bit more, let's say, stringent, we use T7 polymerase based promoters and T7 polymerase as the polymerase which is responsible for transcription, because this promoter, this system, let's say its control is tighter and you have less background. In our case, the background is not a problem, but sometimes we are producing toxic products, toxic proteins, and not only because of their activity, but also because of their structure. So they're, for example, difficult to be folded. They yield inclusion bodies, and bacteria suffer because there's so much of insoluble material. And sometimes even the structure of the mRNA or the content of mRNA can be inducing bacterial cell death. And there are also, for example, bacterial strains where you can prevent that. Okay, so production of a new protein, if you want to express a gene and produce a protein, this is always a biological question. Will it work? How it will go? You have to always check it first experimentally. You have to check the solubility of your protein, like we will be also doing this week. So we were testing the solubility of our GST fusion protein, and you will see that it's partially soluble. It's not 100% soluble. If you want a protein which is 100% soluble, this will be GST only. If you want to have pure GST, this is really soluble. Generally, GST tag should increase protein solubility. This is one of the reasons why we are using such a big protein tag, especially for small proteins. But still, we may end up with a protein which is only partially soluble. Partially soluble is good enough, because as long as you can get nice protein yield, and we need usually a protein in the soluble fraction. There are different types of resins, and in some of them you can also use denaturing conditions. So you can use detergents and denaturing agents, and you can purify your protein which is denatured, so it's not actually folded properly. And the best resin for that is Histag. So Histag and nickel or it's nickel or cobalt based resin for purification. For GST, we cannot use denaturing conditions, so we have to have our protein in the soluble fraction. Can you tell me why? Is it so? Why we cannot use denaturing conditions for GST fusion proteins? Can you think about it? So we have our GST CD81 level. Why it has to be soluble? So folded properly.
The protein is too complex to be denatured and renatured again?
Not really. GST, I think it can be renatured quite efficiently. It's not that bad. It's the idea of the beads. So we are using affinity chromatography, and we are using an enzyme and its ligand. So what we know about enzymes is that usually they have to have some kind of a conformation to recognize the ligand. So if we want GST to recognize glutathione, we have to have it in the right conformation. That's why we cannot denature this protein. It will not bind GST resin. Generally, that's it. So when we want to use affinity chromatography, like for example, we are purifying antibodies using protein G, cepharose, or antibody conjugated resins. When you want to just use antibodies to fish out your protein, then the structure is very important, and you cannot denature your protein, for example, with the guanidine salts, because usually guanidine salts are used to denature your protein. Okay, and as regards bacterial strains, again, there is a variety that you can buy and you can use. You can try different ones and see which one gives the optimum yield, and they differ. Our strain, as I think already mentioned, DH5 alpha, this is not maybe a recommended strain. It contains proteases, but still, I think for our purposes, it works a bit better than the standard BL21 based strains. So this is, let's say, the first type of strains. They are BL21. and you have DE3 in brackets. Do you remember from molecular biology what does it mean that you have DE3 in brackets? What's DE3? I mean, it's stated there in this table, but maybe you can remember what it means. Each time when you will see a bacteria and you will have, well, usually there's DE3 in brackets because this is the strain of a virus which is used. So this is lambda phage. And this particular lambda phage, this is a recombinant phage, which is carrying T7 polymerase gene. So it has a gene from a different bacteriophage, T7, encoding its RNA polymerase, and it's bringing it to E. coli strain. BL21 is derived from the B strain of E. coli. What we can say about those strains, of course, they are not pathogenic. They grow a little bit slowly, and this is also good for expression. Generally, slower growers of bacteria give less background. And what's also interesting, those bacteria, they do modify DNA. So, for example, we cannot use it to propagate plasmids for molecular biology because they will be methylated, and as you maybe remember, some restriction enzymes are sensitive to methylation. So if you want to produce DNA, you will not use those strains. Then we use methylation deficient strains. Okay. So when we have T7 polymerase, we are using plasmids with T7 polymerase promoters, and this is a kind of a stringent expression. The background is lower. The expression of T7 polymerase is based on lac promoter and is induced by IPTG, but it's like two-step control. Okay, you induce T7 polymerase, and T7 polymerase must bind T7 promoters, and then you have expression. And then we have modification by a second plasmid, which is called PLYSS, so PLYS. It's encoding additional T7 bacteriophage lysozyme. And it's not for the lysis of bacterial cell walls, but this is a control of the T7 polymerase. So T7 polymerase can be also controlled by this lysozyme. So it's also reducing the background, okay? So that it just destroys the T7 polymerase before induction, if there is any background basal expression. Okay, other problems that bacteria face. So there are also strains when you have tunable expression of this lysozyme. So you may increase its expression, decrease its expression. There is this tuner strain, which is also interesting because it's encoding lac permease, and this changes the bacterial cell walls so that IPTG gets to the bacteria at the same time. So it's not slowly incorporated to bacteria for the induction, but it's like happening at one time. If you want to have like, you know, set induction at one time point. Another problem we have with the folding, protein folding, generally recombinant protein folding, especially with the proteins which have lots of cysteine bonds. GST has lots of cysteine bonds, so, but still, its folding is not that difficult. But if you have your protein of interest with lots of cysteines and there are cysteine bonds required, they may end up in inclusion in the inclusion bodies just because the bacterial oxidoreductases, they are not that efficient. And then you have strains which are called origami. Origami like folding of paper, here folding of protein. It has expression of two genes. One is thiooxidoreductase and the other is glutathione reductase. and they are helping recombinant proteins to fold. Actually, we tried this strain once with GST CD81-LEL, and we compared it to DH5 alpha, and there was no difference. So apparently for this protein, it doesn't help that much. It was not more soluble, but you can try that. And then there are strains which are called Rosetta, like Rosetta Stone. Another protein problem with foreign proteins introduced to E. coli is the accessibility of tRNA and something which is called codon bias. So if you remember from our molecular biology, is that each organism has its own set of tRNA which are used more frequently than the others for the especially degenerated codons. And so tRNA which are used in bacteria may be totally different. I mean, the frequency may be totally different than in humans. So human proteins in bacteria may not be efficiently expressed, and also bacterial proteins, if you want to express them in eukaryotic systems, they may not be efficiently expressed just because there is not enough of tRNA. And Rosetta have overexpression of some tRNAs, so-called rare tRNAs, okay? There are also different variations of those strains. There is a strain which is called Rosetta Gummy, so it's like Rosetta plus origami mutations, changes, which can yield better proteins, okay? Generally, you can try different ones. A popular mutation in the majority of the expression strains for purification is the deficiency in proteases, in LON and OMP-T proteases. Okay, so they don't have those main bacterial proteases, and they should not degrade your protein of interest. Anyway, it's recommended to use protease inhibitors, like you will be also adding to your cell preparation. Okay, so these are strains. Do you have any questions to that part? So generally, some things we just try to predict, like we buy special strains if we expect any problems, but it is true that we just have to check experimentally which strain is the best for our protein. Okay, so now I think we can finish this one. And again, I will just switch to the other one. Before we start, I just want to ask you whether you understand everything from your laboratory classes so far. So, do you know why we were purifying lysozyme? The first question.
Because we are going to use it later for bacterial lysis.
Okay. We want to use it for bacterial lysase, and I think I already told you that if you buy lysozyme commercially, we will have a commercial preparation also that we can just add. So it's also, we purchase it as a powder. And it says, this is chicken egg lysozyme purified by CM Cefatex, exactly like you are doing. So I want to ask you, what was your yield of lysozyme? So do you remember, do you know the concentration that you got for the best fraction, concentrated fraction if you are concentrating it? So do you know your concentration? You will have to put it in your report, so it's good to know it. So group 5, what's your lysozyme yield? Silence. So have you measured?
I don't have it calculated directly, but from looking at the calibration curve, I can guess it's around 0.2 milligrams per milliliter, if that was the
okay. And
units
have you
that we were...
measured the protein concentration by spectrophotometer?
Yes, it's from the readout from the array when we had the calibration
Yeah, but
curve.
this was by BCA kit or
Yes, the BCA.
activity. And what about the UV based spectrophotometry?
I think it was meant for the next lesson, or I don't really see it.
Okay, so maybe you skipped that part.
Yeah, I think I skipped it.
You skipped it. So group 6 did not skip that part. So what's the concentration of lysozyme from group 6?
I think it was around 0.9 milligrams per milliliter.
Yeah, right. So this is our concentration, and this is after concentrating the lysozyme. So the yield was not great. And I already tried to explain that we are just unlucky because we are using the same resin. And apparently it was not washed properly. So maybe we just had some groups which were not activated enough. And our yield generally was just lower. You could see it from the activity of the fractions. So because we need 0.2 milligrams per mil as a working concentration of lysozyme, you will have to add some commercial preparation, which is 20 milligrams per ml. So generally that's it. Okay. Were there any problems that you encountered during the classes? Something that you don't understand? So as regards the elution graphs, please remember that we need all the elution fractions to be included. And you do include this first two big washing fractions, like big 1 and big 2, because we want to be sure that if there was any lysozyme inside. So did we had all the lysozyme bound to the resin, or maybe we just, let's say, we just overloaded our resin with the sample, so not all the lysozyme could bind. And you do not include in your graphs the starting fraction, so this W, egg preparation. You don't need that, this is not elution, okay? But all the other fractions are there. Okay.
So, you mean the activity or the concentration?
You are putting both on the same graph.
Yes, but for the concentration with the BCA kit, we only tested like the last 4, or the ones that were left. Okay, we didn't measure from 1 to 10. We just picked the last one. That's what we were told. We got the activity tested for everyone, but the concentration was tested only for the last samples.
Okay.
And the first two where the lysozyme was still in the column, the big 50 milliliter ones.
Okay. And which fraction had the maximum activity of the lysozyme? Those last 4?
Yes.
Oh, okay, so that was the reason. Okay. Yeah. Yeah. What can I say? It should be measured for the all fractions so that you know how it goes. So, for example, that you understand that you have lots of proteins, for example, coming in those big 2 fractions, or at the beginning of your elution, because we have, of course, different type of positively charged proteins in our sample, not only lysozyme, and they can be bound, but because they differ in their PI, they may be eluted also in different fractions. So, okay, yeah, but if you did it like that, then you just put those last 4 fractions for the graph. What can we say? Okay, so I think we already mentioned what is ion exchanger and what's the isoelectric point of a protein. So we can just try that. So this is in Polish and this is in English. You should know what are the most popular active groups which you can use in ion exchange chromatography. And generally, if you are like preparing for protein purification, you should choose your resin. And the choice is really broad. And sometimes there are kits, like sets of different resins that you can just try. You just check which one is most optimal for your protein. Like you get 4 columns with different types of active groups, and then you check and you know that this one is the best. So generally when we are buying our resins, they have different commercial names, like it is stated in the table. It can be CM Cefadex C25 or Q Cefarose FF. And we should understand, but sometimes the names are even less informative, like Biogel something something. And of course we should understand what it means. So first of all, we have different types of a support. So our active chemical groups are just chemically bound to a support. And so you have a picture of from the electron microscopy, so that you can know that it's really porous. And usually we are using Cefadex. I think already mentioned that this is a modified dextran. Then you have Cefacel, which is modified granular cellulose. Cefacryl will be, or Biogel, very often Biogel with P. This is also acrylamide. And Cefarose, this is a trade name for agarose from one of the companies, or Biogel A. Generally, some supports are more porous than the other ones, like our Cefadex. So it helps a little bit more because it also works like a size exclusion chromatography. Smaller proteins just go into the resin where they interact with the active groups. So they differ also by that. When you are using Cefadex, there will be usually C with a number. C is like cutoff. And the number just is telling you what's the size of the pores. And it's either recommended for smaller proteins or for bigger proteins. Like C25 is for smaller proteins, and you may have just the cutoffs are different. Okay, so like our cutoff I think is from 0 to 30 kilodaltons, and so it should be better for smaller proteins. Sometimes you have, especially with modified cellulose, you have G with a number. G is like from granular. Okay, and then you have your active groups, and they can be positively charged or negatively charged. And depending also on their pK value, they are regarded as strong ion exchanger or weak ion exchangers. And generally, the PI, so the isoelectric point of a protein, decides whether you can use a strong ion exchanger or weak ion exchanger. Okay, if the PI of your protein is around neutral, like a little bit acidic or a little bit basic, and you cannot use also harsh conditions like really extreme pH conditions, then it will be difficult to use strong ion exchangers. Then you just go for weak ones. But generally, strong ion exchangers should give you more pure protein. Okay? So, the most popular groups, I think I also stated them here in the English version. So we have sulfur groups, or sulfomethyl or sulfopropyl groups, which are negatively charged. And this is usually a strong cation exchanger, so it binds positively charged proteins. And usually you use, let's say, more extreme pH range buffers just to elute the protein. So that's why it's also tricky with GST, or tricky with lysozyme if you want an enzyme. We are using carboxymethyl group, which is also negatively charged, and it's binding cations, but it's a weak ion exchanger. So you may use also different type of buffers. And then we have, in the, let's say, opposite direction, we have anion exchangers, positively charged groups, which are usually secondary, tertiary, or quaternary amine groups. Like we have Q, the strongest anion exchanger. It has quaternary amine group. You have 3-ethyl aminoethyl or d-ethyl aminoethyl, which are tertiary or secondary amine groups. So Q is quite popular for anion exchangers, as if you have proteins with low pi, and DAEA, the ethyl aminoethyl, for example, is very popular to purify DNA. If we are purifying DNA, we have those different types of kits. In the majority of, let's say, cheaper kits, you will have just silica to bind DNA. But sometimes the kit just says that this is AI, yeah? AI, ion exchange. And then it will be DAEA, so diethyl aminoethyl, almost for sure. This is used for nucleic acid purification. Okay, so we have resin, this is one part, and your buffer, this is the second part. So you also have to choose your buffer. And it all depends on your protein. So whether it has enzymatic activity we want to preserve, whether you want to have some organic compounds inside or not. So this is the decision you can just follow. Also, if a protein has been already purified by other people and the results are published, you can also follow the conditions. Which buffers are very popular for lower pH? We have citrate buffers. We have phosphate buffers sometimes, and I think this is quite popular. And as regards higher pH, well, we have carbonate, but very often we have just trees-based buffers. And yeah, they can be also quite popular as regards higher pH. Okay. So I think this is all about ion exchange chromatography, just as a reminder what you already had. Just remember what's the isoelectric point of a protein and what happens to a protein when the pH is higher than the pi or lower than the pi, not to mix which groups are protonated and which groups hydrolyze. Western blotting. So this is something that we will do this week because we are checking the solubility of a protein. And this is usually done when you are doing purification for the first time. If you're not sure whether your protein gets into the soluble fraction, it doesn't make sense to purify it, of course, because you can just waste your resin. Well, many resins are reusable. You can just use them for many times. But why should you do that if your protein is insoluble? So we check it by Western blotting because we want to be sure which protein is our fusion protein. And what we should remember about Western blotting. Yeah, well, first of all, that there are many steps when we can make mistakes. And these are the types of Western blotting, so the results that we may have. We will see how it goes. And so now lots of questions, and I would like now also other people to share their knowledge. So this is just to remember, and you should know at least this this system. So SDS page, standard SDS page, which is discontinuous reducing Lamley. Okay, so why is it called discontinuous, and of course why is it called reducing in the presence of SDS? So why is it called discontinuous electrophoresis?
Because we have the resolving gel and the separation gel? Yes?
Yes, just because we have those different pH conditions. Okay. And why is it called reducing? Other people?
So we have SDS, which is detergent, and it's reducing protein or beta mercaptophenol.
Yeah, but this is the reducing of the disulfide bonds. So that's why it's reducing. And it's denaturing, and denaturing is because of SDS mainly. Okay, this is the main denaturing agent that we are using. Okay, and why is it called Lamley? Very often you will see that this is Lamley buffer or Lamley system. What does it mean, Lamley here? Guess.
Because of the person who discovered it or improved it?
Yeah, this is the name of the person, the guy that invented the system. Like we have, for example, for semi-dry blotting, we have Taubin buffer, and Taubin is also a name for the person. Okay, so traces, different pH conditions, and we have to understand what's going on. So why we have isoelectric focusing of the protein sample in the upper gel, stacking gel, and why do we have resolving gel with different pH? So now please explain to me what's going on there. So generally, what's the pH influencing?
The charge of the protein.
The charge of the protein, a little bit, yes, but this is not the whole, this is not the idea here.
It's because of charge of glycine.
This is the charge of the glycine that it's affecting mainly. This is what we want to do. So what's so specific about glycine?
Its PI is about 8.8, I think, but I'm not sure.
Oh, it's lower.
It's a little bit lower, maybe. That's right.
I think somebody checked, it's like 6.3 or something. So it's a little bit below neutral. And it's quite close to the neutral pH. But that's why it's nicely tunable, okay? Because you can go in this direction or in this direction. And in the 6.8, it's a little bit negatively charged, a little bit, but it's almost neutral. So what's now the order of the compounds separation in the stacking gel? How does it go? So what's at the head of the sample? What's going first?
On the head is chlorine ions.
Yeah.
Then proteins, and then glycine, and the electric field just packed our our proteins and allows to go into the running gel in one time.
Exactly. So we should remember that there is also electron movement because of those compounds, and this is actually stacking our sample. And chlorine ions because they are negatively charged small ions. So then resolution, so resolving gel, how does it work?
The glycine gets a much more ionic change, let's say.
Yeah,
and it's
negatively
more,
now?
yeah, that's right. And it's small and just go before proteins and goes with chlorine ions on the...
Yeah, and then our proteins resolve based on their molecular weight because the charge is neutralized. Well, not neutralized, but it's, let's say, set to negative by SDS. Plus, we have to remember that our polyacrylamide is also working as a kind of a sieve. Okay, so it's also helping to resolve the protein because proteins may still have a bit different different structure a little bit, like some may be more fibrillar, and it still helps to resolves. Okay, acrylamide, bisacrylamide. Why those two compounds?
You mean that we can use either one? Yes, this or this?
Well, generally by changing the concentration of bisacrylamide, we can make the
changes
pores.
in the pores that are making.
Yeah, we just change the pores, so they can be bigger or smaller. And protein acrylamide concentration depends on the protein size, and I think we will show. I will show you a small tables how you can, let's say, choose the percentage of acrylamide for your electrophoresis. Okay, then we have also other molecules like SDS, and I think that's quite clear. Everybody remembers that this is an ionic detergent. It's negatively charged and it's wrapping your protein, and it's also denaturing because it's destroying quaternary and even tertiary structures, or even secondary a little bit. So it's like actually denaturing and giving charge. And then we have bromophenyl blue in your sample buffer. So your sample buffer is blue. So what's the role of bromophenyl blue?
It just shows us where the gel is currently at.
Okay, why?
Because we need to know when to end the electrophoresis to not let our samples out of the gel.
Okay, but why is it showing us?
Because it's blue.
Because it's small. It's a small dye which is not binding the proteins, which is not binding the gel. It's just moving at the head of the sample because it's small and negatively charged. Negatively charged. Okay, APS and TEMET, so ammonium persulfate and tetramethylene diamine, ethyl diamine. So what's the role of them?
APS is the source of free radicals, and TEMET catalyze formation of them.
Okay. Okay. And then boiling. You boil your sample. So why are we boiling our samples?
To help it denature?
To denature the proteins, yes. So we have SDS denaturation and we have heat-based denaturation. And please remember that some proteins cannot be boiled, especially if they have structures sensitive to this type of denaturation, and they may form aggregates, big aggregates, so that they will just stay in your wells of a gel. They will just not get into the gel at all. You will see empty lanes, for example, if you do Western blotting. So this is, let's say, checked experimentally. So if you don't see the band of your protein in your Western blotting, this is one of the reasons you might think about that your protein just cannot be boiled. And then you can heat it. You heat at elevated temperatures like 65, 56, and sometimes even 37 degrees, because they cannot be heated. Okay, so then there is a question, what else is present in the sample buffer? So we have, for sure, trees. In the sample buffer, there is still SDS added in the sample buffer. So you have SDS in the gel and you have SDS in the sample buffer. And then there is bromophenol blue. And what else?
Glycerol.
Glycine.
Glycerin.
Glycerol. Yes, yeah. Glycerol. Or it can be also sucrose. So it depends. I think sucrose for proteins is more popular. Generally something with high density viscose, so it keeps the sample in the wells. Otherwise, it would have a problem with just staying in your wells. It's like with DNA, we add glycerol to the buffer, and here usually sucrose to the buffer, so that it's a bit, let's say, heavier. Okay. Generally, this is the idea how the SDS works. Well, it's not that we fully understand, and there was a lot of research on how SDS is actually denaturing the protein, making whether it's totally unfolding the protein to the primary structure or not, and it's not actually producing pure primary structure. And on the right, you have the, let's say, the principle of ammonium persulfate, so the free radicals that it produces and how it works on the polyacrylamide. This process is sensitive to oxidation. That's why we have to use a layer of water, the simplest way, or isobutanol, because it's not reactive, and it's also, the density is proper. I mean, it's lower than of the polyacrylamide, so it can just form a layer above acrylamide and just stop the access of oxygen. So these are models of SDS-bound proteins. So you can see that actually there are small domains which are formed with the micelles of SDS, and that's why each protein can differ a little bit in this, let's say, structure that is formed with SDS. So this is a model, they call it like beads on a string. So you have those small domains, these are beads, and you have a string. Okay, so again, we have stacking in the stacking gel, and we have electrophoresis, so the resolution in the resolving gel. And now the percentage of acrylamide and the separation of your proteins. It also depends on the buffer system. So in our tris-glycine buffer system, that's how it goes. If you want to have your small proteins just to stay in your gel, it must be more concentrated, like 15%, 12. Well, with 12, you're risking, because sometimes, especially when we are preparing the gels ourselves, we don't buy the ready ones. It's still prone to some errors. So it may happen that the smallest proteins, they just get out of the gel. And if you want big proteins to be resolved, you have to use 8% gels. Sometimes people do 6%, but 6% gels are very wobbly. They are very fragile because there is the concentration of acrylamide is quite small. So sometimes you need a special support like silicone coating of the plates so that, let's say, your gel doesn't crack during separation or after separation. We may also use gradient gels. So there is a gradient of polyacrylamide concentration, like from 4 to 20% or 8 to 16. And they are gradient mixers that you can use to prepare a gradient yourself, but usually we just buy ready gels. Or if you want to have really like homemade gradient gels, we just make layers, like a layer of 20%, a layer of 15%, and then like 8% top. It helps if you want to have small proteins and big proteins in the same gel resolved. This is what you can use. It works. It's not maybe totally perfect, but it works. And then you have also other buffer systems for small proteins and peptides. This is trypsin buffer, and then you can go up to 20% of polyacrylamide, and you can separate actually peptides. And this is done for small proteins. And these are also other like mass mops, different salts that you can use for your buffers. And in the commercial buffers that you use, which we buy, usually I think this is mass or mops which is used. So just different type of salt. Trisglycine is, I think, still the cheapest. Okay, Western blotting, and we are supposed to see a movie. I will see whether we can actually do that. Maybe just some kind of a theory. Okay, so this is the machine. Yeah, we should see the movie, I guess. This will be better. I didn't put the movie into the presentation. Because generally, well, usually it seems that you know all of those things, but sometimes the small details are just, let's say, not there. Okay, let's see how it goes. And then again. Yeah, and then again, if I have this presentation, then I have a problem with just closing it. And okay, maybe here. No. Okay, then I think we can just. It's a pity. Why can't I do that?
Sorry, can I ask a question?
Yeah, sure.
It's more about the calendar and organization part of the laboratories.
Yeah.
It would be easier for me to ask it in Polish, so can I?
Okay.
Tak więc ja jestem ogólnie na zwolnieniu chorobowym. Mnie jutro nie będzie na tych takich dłuższych laboratoriach i wie Pani może w jaki sposób ja będę musiał to odrobić?
Każdą sytuację prowadzący rozpatrują indywidualnie, także potrzebne jest na pewno zwolnienie lekarskie.
Mam.
I jeżeli to jest jednorazowa nieobecność, zwykle po prostu nie każemy jej odrabiać. Wszystko zależy od tego, co jest celem ćwiczeń, tak? Także...
Bo ja właśnie jakby nie wiem, czy jutro jest ostatni dzień mojego zwolnienia, bo idę znowu na konsultację do lekarza, więc...
Każda sytuacja jest indywidualna, tak? Ciężko mi jest jakby tutaj jakby z góry przewidzieć, bo po prostu no mamy warunki pandemiczne i nie jesteśmy
dobra.
już tak, że tak powiem, ostrzy, jak to wygląda.
Dobra, dziękuję.
So I'm trying again. Oh, this is not this one. Okay, this should be okay. Okay, so can you see the movie now? Yeah, but it's a Can you see the movie and can you hear it? That's the problem because
We see it.
We can
you
see
hear
the
it?
movie, but
Not really.
not really.
not much.
That's the problem that I have to. I'm sorry. I have to. For the presentation, I need. Okay. And now. Okay, I need my voice. My sound, that's good. And now this one. Okay, so let's get back to the beginning. Okay, so small relax.
Chemiluminescent detection is the method of choice for Western blot analysis because of its signal output, high sensitivity, and versatility for use with film or digital imagers. This approach requires a membrane probed with an antibody that is conjugated to an enzyme, typically horseradish peroxidase or HRP, and a chemiluminescent substrate. These substrates are usually two-component systems, consisting of a stable peroxide solution and a luminol or acridant solution to provide enhanced chemiluminescence, or ECL. While commercial ECL substrate kits vary in sensitivity, they all follow a general protocol. First, combine the luminol or acridant solution and the peroxide at the ratio indicated by the manufacturer. Next, pour the solution onto a membrane probed with an HRP conjugated antibody. The luminol or acridant is oxidized by the peroxide in the presence of the HRP enzyme, resulting in the emission of chemiluminescent light. After incubating the membrane with the substrate for 3 to 5 minutes, remove excess substrate and wrap the blot in plastic to prevent it from drying out. Let's develop this blot with film in a darkroom. Under a safety light, expose a piece of X-ray film to the membrane for a specific length of time, which must be empirically determined. When the time is up, process the film according to the instructions of your film developer. If the band intensities are too light or dark, or if the background is high, expose a new piece of film to the blot and increase or reduce the exposure time. Digital imagers are rapidly replacing the use of film for Western blot detection because they are more sensitive and have a wider dynamic range of detection than film. These instruments use a CCD camera to capture the chemiluminescent image of a blot at preset and custom exposure times. To develop a blot using an imager, place the blot onto the image platform and select the exposure time. Band intensities can be adjusted simply by selecting another exposure time. Also, imagers often come with software to help you determine the best exposure time for your blot. Developing blots is faster with digital imagers than with film because you don't need to wait for up to 5 minutes for the film to develop. And because your blot is already digital, you don't have to scan the blot to perform image analysis or to include it in your manuscript.
Okay. Okay. Okay, getting back. Sorry, this was commercial, but generally it was supposed just to show you what's the old traditional way of using just X-ray films to develop a Western blotting using chemiluminescence. Now, of course, we have nice digital imagers, but still, if they get all broken, you don't have to stop your experiments. We have dark rooms also in the building, so you can just use this. This is like dentistic X-ray film. It's not the standard one that we are using. Okay. And now again, I just want my presentation. Okay, two types of transfer of proteins to a membrane that we can use. This is wet transfer or semi-dry transfer. We don't have dry transfers, of course. And even the semi-dry transfers, they are not that dry. I mean, you cannot really let your sample to dry. And the question is when we are deciding that we want to use wet or semi-dry. And the answer is generally that it depends on time, usually. So semi-dry blotting is much faster, because now if you're using commercial reagents, you can do it in 7 minutes. That's at least what the company promises. But usually we don't go beyond 1 hour, even with big membranes. But if your experiment is quite long and you end up in the evening and you don't want to stay for this extra hour, you can set up the wet transfer overnight, just go home, and in the morning you will get your proteins on the membrane. So there are like pluses and minuses of each method, and it usually depends on the decision of the researcher. The membranes that we are using to transfer your proteins, so get a replica of a gel, basically there are two types. And we are using PFDF, which is polyvinylidene fluoride. This is hydrophobic membrane, and it's binding the proteins, and you can get them back from the membrane as well. That's one of the reasons. And there are also different types of pores of the membrane. For example, for small proteins and peptides, with water or buffer. Whereas with PFDF, you have to make it wet using alcohol, like methanol usually. And nitrocellulose will dissolve in methanol. So if you mix those two, you may end up with a just damaged membrane. Okay, and for the transfer, we use the same, not the same, but a buffer which also contains tris and glycine and SDS. There is methanol added to increase the generally heat capacity of the buffer, because there may be high protein, just high current, which is just going through the sample, in especially semi-dry blotting. So semi-dry blotting buffers are always with the methanol, just to prevent heat from damaging your membrane, even though sometimes it happens that you get like yellowish membrane after the transfer. Okay, and the conditions of the transfer, of course, we follow the instructions of the producer, but sometimes it just has to be established experimentally. With the semi-dry blotting, the problem is that when we open the sandwich, like we open the machine, the blotter, and sometimes our sandwich separates and it's impossible to put it back together just to, for example, prolong the blotting. So that's the only problem. And then Western blotting, this is the incubation with antibodies. So this is detection with antibodies. Usually, this is a two-step incubation. We have primary antibodies and secondary antibodies. This is usually because of money. Just buying conjugated antibodies directly with an enzyme, it's a bit more expensive because unconjugated primary antibodies can be used for different methods. Generally, that's it. So we have primary antibodies, and we have to have a blocking reagent because, of course, on our membrane, there are bands of proteins, but there are also empty spaces. And our membrane likes to attach proteins. So, for example, if we just touch it with our fingers without gloves, we may have our fingertips, let's say, marks blotted as well. I mean, detected with some type of unspecific reaction. And for blotting, we are using semi-dry milk, and this is a commercial milk. And in every country, this is a commercial milk. So I think the companies, they don't produce the milk, skimmed milk for blotting. They produce other reagents. So we just buy it in shops. And the only condition is that it has to be low-fat milk. So skimmed milk, actually, because fat residues may affect your blotting. The company, the brand of the milk may also matter. So like we have our favorite ones. And I think this is all over the world. For example, when I was working abroad, I could see the skimmed milk from one of the popular companies, which was generally producing dairy products. So then it was also producing the skimmed milk, and it was used for Western blotting there. So it's not only Poland, this is all over the world. Sometimes we cannot use milk. And can you think about the situation when we cannot use milk as a blocking reagent? For
Maybe
example,
if our protein has an affinity to some proteins in milk?
Well, our protein will not like jump from, but our antibodies may have affinity to milk proteins. For example, sometimes for histacked proteins, in milk there are proteins with polyhistidine, and they are like recognized by antibodies. Sometimes, some antibodies. But generally, for example, for histacked detection, milk is not recommended as the blocking agent. So you will just know it experimentally. If you get a black membrane with high background signal, it may mean that your antibodies recognize some milk proteins, and you cannot use it as a blocking agent. Alternative, you may use just single milk proteins, like people are using BSA, bovine serum albumin, or casein, for example. And there are also artificial chemical compounds like blotto, for example. So they are just chemicals which are blocking the membrane. But blocking is quite important. We have to do it, and for just the blocking step, usually we use at least 3% of the skimmed milk. You can go up to 6%, and you can do it for 1 hour. You can do it overnight. Usually no longer, because generally this is not sterile. And if you just use milk for a longer period at elevated temperatures, the bacteria will grow inside. So your milk will just fermentate. You will see that. So it's better, if you want to keep the membrane wet, to keep it in the buffer, in the TBST buffer. Then you can keep it longer. It can be also frozen, but then without liquid. So you can freeze it in a bag and just, for example, detect another protein on the same membrane later on. Okay, Secondary antibodies. Actually, so the detection system, the secondary antibodies which are recognizing the primary antibodies, they should not be cross-reacting. So they should recognize only, for example, mouse antibodies or rabbit antibodies, no bovine proteins, like bovine serum proteins, milk proteins. And they are conjugated to an enzyme. And there are two types of enzymes which you can meet. One is alkaline phosphatase. This is a bacterial E. coli alkaline phosphatase, or horseradish peroxidase from horseradish. And it all depends on the substrates that you want to use. So you can use either colorimetric method or you can use chemiluminescence. Well, horseradish peroxidase can be also used with colorimetric method. There are also substrates for it, but usually then you have less sensitivity, just lower signal. So horseradish peroxidase goes well with chemiluminescence, and alkaline phosphatase with the colorimetric method. There are substrates for chemiluminescence for alkaline phosphatase, but they are quite expensive. Generally, AP is more sensitive than horseradish peroxidase, but not with the chemiluminescence detection. For chemiluminescence detection, you need X-ray films, like we show in the movie, or so-called phosphoimagers, or the imaging systems, and you have two different ones, like Biorad, which is in the building. We have UV-Tech system, which is in the building, and they are different brands. And they differ in many aspects, but generally each of them should work for a simple blotting. Okay. The imaging systems can be also used for antibodies which are fluorescently labeled. So this is, let's say, the most recent achievement. We don't use an enzyme linked to antibodies. We use fluorophores, and we can, for example, stain proteins with two different antibodies with two different colors, like here marker is red and the protein of interest is green. But then the imaging systems have to have special filters. Not every imaging system can detect this fluorometric signal. And then the sensitivity will be also quite high. Okay, so this is all in the slides. With chemiluminescence, there are different substrates that we can use, which differ in the concentration of luminol and its stability. And as a result, they differ in their sensitivity. So how much of a protein they can detect. And now it all depends on what you expect. If we are having a protein like we have, purified protein, high amounts, low background, then you don't need very sensitive substrates for the detection because your background will be much higher. So you will get a ladder of a protein instead of a single band. Then you choose less sensitive substrates. But if you're detecting a protein which is not abundant in your sample, then you either use lots of material for your gel, or you choose more sensitive substrate. For example, there is luminol stabilized by coumaric acid, then it's much more sensitive. And we should all remember that the real substrate for horseradish peroxidase is the peroxide. This is the substrate actually. So luminol is like a bystander reaction. Okay, so we use something which is produced from the peroxide that just reacts with the luminol and changes the luminol so that it produces light. Okay, so I think this is all for Western blotting. So the question is, if you have questions. So just think about them. I hope our Western blottings just go well, so the transfer is sufficient. How we can, for example, estimate the efficiency of our transfer. So, of course, there are at least two ways. First, or three ways that I can think of. So first, we are doing blotting. Usually we are using a press paint marker. So you have colorful bands, and you just see that those colorful bands are transferred onto your membrane, and you can see whether they stay in the gel or not. Usually smaller proteins are transferred with better efficiency, and bigger proteins may stay in the gel. If you don't see the proteins remaining in a gel, you can still stain your gel, like with Kumasi blue, and check whether there are any proteins left in the gel. You can also stain your membrane, but some staining methods may interfere with your Western blotting. So usually we're using reversible staining, and there are like two dyes that are commonly used. One is Ponceau S. So this is red dye, which can bind proteins and can be washed out with water later on. So you just stain the proteins and then you destain with water. And there is, I think, also amido black. So it's a blue dye which can, depending on the solvent, irreversibly or reversibly stain your proteins. And then you can also check whether they are actually on the membrane and whether, for example, there are equal protein contents in your lanes. For example, it may happen that like right part of your blot will transfer better than the left part of your blot because, I don't know, something got swayed and, well, it just happens from time to time. So generally that's it as regards your Western blotting. And so the last thing I just wanted to show is this part of experiment that you will not see just because of the time frames. But actually you could do it because you're starting a little bit later. So this is the preparation of the bacteria for lysis. So the DH5 alpha, which are overexpressing GST CD81 level. So if we do this experiment properly, like from A to Z, we would start from preparing competent cells, bacterial cells. You can either buy competent cells or you make them competent yourself using various methods like chloride, a calcium chloride method, or there is like a Rubit method what you can use. Generally, you need them competent, like for molecular cloning techniques. And then you transform them with a plasmid. PJECs in case of GST fusion proteins. They are plated on a plate and they form single bacterial colonies. And then from a single colony, you start a small volume overnight culture. And when you have this culture, like we have here on the left, there is a small flask. I don't know if you can see it. The flasks which are around, they have just LB medium, and the one which is in the middle, it's full of bacteria. We use LB with ampicillin because our plasmid is encoding beta-lactamase, so a resistance to ampicillin, and the bacteria do grow. And then we have to refresh our culture. So if you want to be really up to the book, you refresh it 1 to 100 volume ratio, and we can use 1 to 50 if we are in a hurry, like I did for you. And we just start it in fresh flasks with fresh medium. And then you put it to a shaker bath, either dry bath like we have here, so just incubator, which is shaking the bacterial cultures, or there is a wet bath, so it's filled with water and keeps 37 degrees, and the bacteria are rotated at 200 rounds per minute. usually. So like 200 RPM, this is quite fast. We shake them because E. coli, this is not an anaerobic bacteria. It requires some oxygen, so we have to provide oxygen at least by mixing. This helps. And then we have to grow the culture for a proper optical density, OD, and in the instructions you have a range from 0.4 to 0.6, which is logarithmic phase, and we already know that it must be logarithmic because this is like the best fit bacteria for protein production. Usually higher densities are a bit better than lower densities, and usually it takes around 2 hours to get the right OD. And for us, it was 0.4, so like the bottom line. And then we add IPTG for induction. And the concentration can be up to 1 millimolar. It can be 0.5 millimolar or 0.1 millimolar, for example, if you want to have overnight induction, and 1 millimolar if this is quite a standard one. And then you culture for the desired time. It can be 1 hour, 2 hours, 3 hours. I did for 5 hours, which is a bit long. I hope the protein will be still soluble. For some proteins, we just grow them in the bacteria in a bit lower temperatures, like not in 37 degrees, but in 30 degrees. They grow a little bit more slowly, and it just helps the proteins to fold a little bit better. So if you have a problem with folding, usually we try overnight culture at lower temperatures, and it can help. So after that, I just centrifuged the bacteria, and you have a pellet ready. It's just a simple pellet from the bacteria, so the supernatant was removed, and your bacteria pellet is frozen. This is a genetically modified microorganism, just to remind you, so we have to pay attention to the leftovers of the glassware and of the culture. We have to kill the leftovers of the bacteria, not to allow it to be released to the environment. Okay, so are there any questions that you have about the lab? You will use different compounds for the lysis, so please pay attention why you need magnesium chloride, why do you need DNase, why Triton X100, what are your beads for affinity chromatography, what are we using for elution later on, but this will be next week, so not this week. I told you that our promoter is a bit leaky. So the best control, negative control for the Western blotting will be empty DH5 alpha bacteria. Sometimes we use the bacteria before induction, like time point 0, before addition of IPTG, but then you will notice some basal expression, for sure, always. Therefore, it's the best just to use empty bacteria to compare if you want to see where is your protein of interest. You will know it from the protein size, but if you get many bands and you want to check which are specific, which are not specific, then you have to have proper controls. So this is what you will use, and you will check the soluble fraction, so the lysate of the bacteria, and also the leftovers of the membranes and inclusion bodies. So usually if we have the protein in the insoluble material, this will be inclusion bodies, not membranes, but just inclusion bodies. And I think the only really soluble GST fusion protein that I saw was just pure GST protein. And this one is not that bad. And for some of them, you just get insoluble proteins still, even if this is GST. Okay, are there any questions? Or questions what you want to put in your report, for example? If not, if everything is clear, then we will end for today. And for the last seminar, I want you to refresh your knowledge on protein tags. different protein tags. We will talk a little bit about affinity chromatography and think about all the questions that you have about your report. If you have any doubts, what to put there, then we can think together. If not, then thank you very much, and see you in the lab. Okay?
Thank you.
Bye.
Thank you very much.
Goodbye.
Goodbye.
Thank you. Bye.
Bye.
Thank you. Goodbye.
Thank you. Goodbye.
Bye-bye.
Thank you.
Oh, excuse me.
Yes?
I wanted to ask you because during the last lesson you showed us the